The overall goal of this molecular cytogenic method is to observe inter-chromosomal stable aberrations in the bone marrow cells of mice exposed to total body irradiation.
This method can help answer the key question in the radiation biology field, such as the risk of inducing stable chromosomal aberrations in the bone marrow cells of mice exposed to total body irradiation. The main advantage of this technique is that it allows direct visualization of radiation-induced stable genetic damages in the bone marrow cells of mice that can be propagated through many cell generations.
After isolating bone marrow from mouse femur and tibia bones and making a single cell suspension according to the text protocol, carefully overlay the cell suspension on an equal volume of bone marrow mononuclear cell separation medium. Centrifuge the gradient at 400 times G at room temperature for 30 minutes. Then, carefully collect the buffy coat without disturbing the remaining layers and transfer the solution into a new 15 ml centrifuge tube. To prepare metaphase cell spreads, add 10 ml of PBS to the tube containing the buffy coat. Then centrifuge the tube at 400 times G at room temperature for 5 minutes. Next, carefully remove the supernatant and gently tap the tube to break up the cell pellet. Then, add 10 ml of PBS and centrifuge the suspension again.